Now moving on to our next question. Question number 6 says, the graph shows the effect of temperature and pH on enzyme activity, which statement explains the enzyme activity at the point shown. Okay, so here you've got Q, P, R, S. So these are the different points here, pH and temperature on the x-axis. So let's read the statements. It says at P, hydrogen bonds are being formed between enzyme and substrate. Okay, this is actually true because the rate of reaction is still continuing, so some amount of hydrogen bonds are obviously being formed between the enzyme and substrate at this point. Okay, so A is the correct response, but what happens is that I've seen many people getting confused with the other options. For instance, why is option B not the correct one? See, let's see. So at option B it says at Q the kinetic energy of the enzyme and substrate are highest. So this is actually wrong because the kinetic energy is directly proportional to the temperature. Okay, or more like as the temperature increases, the kinetic energy also increases. So as you can see here that the temperature still keeps on increasing even after Q. So the kinetic energy at these points will be even higher than at Q. So the kinetic energy is not the highest at Q, but as the highest temperature, the kinetic energy is the highest. So that's why option B is wrong. Let's see why option C is wrong. It says at R, the peptide bonds in the enzymes begin to break. That's obviously wrong because at R there is a maximum activity, not the denaturing point or anything. So then at option D it says the at S the substrate is completely denatured. No, this is also wrong because it says the substrate is completely denatured. The substrate is not denatured, the enzyme is denatured, so D is also wrong. So A is the only correct option, as you can see. Okay, let's move on to the next question. Question number 7, another graph question. So as I've already said, there are many graph questions in this chapter. Okay, 2 enzyme experiments were carried out. The first experiment X was carried out at normal constant temperature of 37 degrees Celsius. In the second experiment, the temperature was increased from 37 to 80 degrees Celsius. Okay, we can see there is an increase in temperature. So which graph shows the results? Okay, very interesting. Okay, so y-axis shows the product concentration, each y-axis, and the x-axis is the time. So we always have to consider the x and y-axis while dealing with this kind of graphical enzyme questions. Anyways, so okay, so 2 enzymes. The first experiment X was carried at a constant temperature. So whenever we carry out an experiment at constant temperature, that means that the product will be formed much more faster. Okay, so the product will be formed at faster rate. So for X, the product will be formed at a faster rate, which can be seen at A. So as you can see, the graph is steeper. That means that this is optimum temperature, so products are being formed at a faster rate and more products are being formed, so there is a higher product concentration is being reached. So B also shows this trend for X. C doesn't. D also shows this trend. Okay, now let's look at the other reaction where it says during the second experiment the temperature was increased up to 80 degrees Celsius. Okay, that's interesting. So once the temperature is increased at 80 degrees Celsius, that means the activity will become very less, almost to the denaturing point, but they don't mention this is where the reaction is going to denature. So there will be, at this temperature, the graph will obviously be a much more less steeper line. Okay, and there will be less product concentration. Okay, so the products will not decrease as it is showing in D. So the products never going to decrease. Like, how can we decrease the products? At B it says the product increases and decreases. The product cannot suddenly increase and decrease because it's a product which we're talking about, the final end product. In C it's showing the same thing as X, but a much more steeper line, as if it's even more. So that's obviously wrong. So eliminating these all other options, only A is the remaining one, where it shows that the product concentration has actually decreased, although there is a bit more steeper part of the graph over here. This is because the temperature is higher, so there will be a bit more higher collisions at that point. but however the product concentration will be less because suddenly the enzyme